Our next session is part of our In the Pipeline series, and this one's on gene therapy. To give us a quick refresher on gene therapy, here once again is Dr. Mena Scavina.
The root cause of Duchenne is a variant or change in the DMD gene that prevents the production of the dystrophin protein. The absence of this protein leads to a disrupted and weak membrane that surrounds muscle cells. One strategy to correct this lack of dystrophin is to deliver a replacement gene to the muscle cells that can function in place of the missing dystrophin. This is called gene therapy. The dystrophin gene is large. Current gene therapy clinical trials in Duchenne are exploring the use of microdystrophins. The microdystrophins are smaller genes than the full-size dystrophin gene. They are based on dystrophin changes seen in patients with milder cases. The hope is that these microdystrophins can act as a replacement for the missing full-size dystrophin and produce the protein that is missing. To understand gene therapy, we need to look at the three main components used to deliver the therapy. First, there is the transgene. This is the instruction for the body to produce a protein. In the case of current Duchenne gene therapy trials, different microdystrophin transgenes are being delivered. Second, there is a promoter sequence. This is a piece of DNA that tells the body where to turn on production of the transgene. The gene can get into many different tissues, not just the muscle. So we want to ensure the new protein is only produced in tissues where there would be a benefit, such as the skeletal or cardiac muscle. Third, there is the vector, which acts as the transporter for the transgene and the promoter to get into the target tissue. In current studies, a modified virus called adeno-associated virus, or AAV, is used. The AAV is a tiny virus with a limited storage capacity, which is why current studies use a microdystrophin transgene rather than full-length dystrophin instructions. There are different serotypes or versions of the virus, such as AAV8, AAV9, and AAVrh74, that are being used in trials. AAV does exist in nature, though it has not been found to be disease-causing in humans. However, because it does exist in nature, it is possible for an individual to have been exposed to AAV and for their immune system to produce antibodies to prevent infection. If an individual has been previously exposed to AAV, they may make antibodies that would prevent the gene therapy from being delivered to their muscle. This could make them ineligible for a trial. The formation of antibodies is also why redosing, receiving a second dose of gene therapy, is currently not feasible. Once someone has been exposed to a high dose of virus at the time of treatment, the immune system will work to produce antibodies to protect them from future infections. Researchers, clinicians, sponsors, and PPMD all recognize this is one of the biggest challenges to ensuring that all individuals can receive gene therapy and be redosed if needed. Many individuals and groups are working to overcome this challenge.
All right, and joining us today for our live panel, we have Luis Rodino-Claypeck from Sarepta Therapeutics, Carl Morris from Solid Biosciences, and Beth Beluccio from Pfizer. As a reminder, we will have a live Q&A after the panelists speak, so if you have any questions, please submit them to the comment box below. So to start the session, we will first hear from Sarepta. So thanks for joining us, Luis. I'm going to hand it over to you.
Great, thank you. Thank you for giving us the opportunity to present today. So thank you to all of the families who are also taking the time to listen, and we're happy to give you an update on our SRP-9001 microdystrophin gene therapy program. If I could have the next slide. This is just a reminder that we'll be making a number of forward-looking statements during this presentation, and these include risks and uncertainties. Please feel free, I'm not going to read this to you, but visit our website to read this in detail. Next slide. In our ongoing SRP 9001 clinical trials, we've now treated over 60 patients, and we have follow-up for some of them past 3 years. So in totality, we have 3 trials. Trial 101 was the first trial, open label trial in 4 patients. This was started with Dr. Jerry Mandel at Nationwide Children's Hospital, and now patients have been followed for 3 years in that study using clinical manufacturing material. Next is trial 102. This is a placebo-controlled trial in 41 patients, where in the first year, half of the patients were treated with SRP 9001 and half received placebo. Now we're in year 2, where the patients that received placebo have now crossed over. And so we're still following the patients in the year 2 of the trial. And now finally, which we'll talk about in more detail today, is trial 103. And this is our first trial using commercially representative processed material. In totality, we have approximately 32 patients planned for this study, and I'll give you more details in the next slide. If I could have the next slide, please. Thank you. So this 103 study is called our Endeavor study. As I mentioned, this is an early stage phase 1b study that's really focused on examining safety and microdystrophin expression of our commercial processed material, or SRP 9001. It's open label, there's no placebo control group. There's no mutations excluded in this trial, and it's based in the United States. So we've started, there are 3 cohorts. Cohort 1 was our first cohort in 20 patients who are ambulatory between the ages of 4 to 7. We've recently added 2 additional cohorts. Cohort 2 has approximately 6 patients who are ambulatory and between the ages of 8 to 18, so older ambulatory patients. Also, cohort 3 is approximately 6 patients who are non-ambulatory with no upper age limit. Importantly, both these cohorts will not require a MIM and washout period from other treatments designed to increase dystrophin expression, but patients will have to stop those treatments prior to the study. As you will see, these cohorts are very small, with approximately 6 patients in each cohort. The primary endpoint for the study in totality is microdystrophin gene expression from baseline to week 12, as measured by Western blot. Next slide. So we've previously shared data from our first 11 patients so far, and we shared this with a PPMD presentation as well. What we found is that safety has been consistent with our previous experience from our clinical material with SRP 9001. In total, we've had 79 treatment adverse events in the first 11 patients, with the most common adverse event being vomiting, which typically occurs in the first week, which is mild and treated with standard medications. We also continue to see increase in liver enzymes that are transient and responsive to steroids, with no signs of impaired liver function. We had SAEs in 2 of the 11 patients that have fully resolved. 1 patient with increased transaminases or liver enzymes who was treated with intravenous steroids, and another patient with nausea and vomiting, and no clinically relevant complement. Next slide. Now, in terms of the biopsies, in our first 11 patients, we performed biopsies at baseline and at 12 weeks in the gastrocnemius muscle. And so, as we've shown previously, we analyzed the biopsies in multiple ways. First, asking the question, did the transgene get to the muscle cells? And so we saw a mean of 3.87 vector genome copies per nucleus across the 11 patients. Next is the protein being made. Are we seeing sufficient quantities of microdystrophin? And indeed, we're seeing 55.4% microdystrophin as compared to normal, mean compared to normal. Then we also ask, is microdystrophin getting to the right place, and how much is getting there? We do this by staining the muscle biopsies using immunofluorescence staining, and then we quantify this in 2 ways. One, looking at the total number of positive fibers, where we see 70.5% in the 11 patients, and then also in the intensity compared to normal. So we see 116.9% as compared to normal at 12 weeks. Next slide, please. So in terms of our clinical development, we now have 2 trials using clinically clinical material from Nationwide Children's Hospital. That was trial 101 and 102. Our first two trials were in the ages of 4 to 7. We're now following patients in the 102 trial during year 2, and we expect to complete part 2 of the study by the end of this year. We're currently, as I just described, in the midst of trial 103, where we have a total of 32 patients planned across 3 different groups, 4 to 7, older ambulatory patients between 8 and 18, and cohort 3, which is non-ambulatory with no age limit. And then we're leveraging all of the data to inform our next studies using our commercial process material, which will be studies 301 and 303, which will both be double-blind placebo-controlled. Next slide. So just to summarize where we are in our development, we've treated over 60 patients with follow-up for 3 years for some of those patients, and we continue our commitment to build a strong foundational knowledge of microdystrophin gene therapy. In terms of immediate next steps, we're going to continue to advance part 2, study 102. This is the crossover phase, and study 103, which is our open label study using commercial process material. We've expanded study 103 to include 2 cohorts. That's patients that are non-ambulatory as well as older ambulatory patients. And we're continuing to do in-depth analysis on all of this using our current data available. In terms of our future clinical development, we're very committed to starting our next study, which is study 301, or our EMBARK study. And our immediate next step for that is to meet with the FDA, and we hope this will occur in the middle of this year. We're using all of our data, including 102 data, to inform the design for that study, and we'll be able to communicate with the community as soon as we meet with the FDA and have more information available on that study. And so just to close out, I'd like to thank all of the participants in our trials, families, the clinicians, and the site staff who are participating in our studies. And I'll be happy to take questions now and at the end. Thank you very much.
One brief follow-up question before we move on to the next panelist. PPMD was proud to fund the original study you termed 101 with nationwide of those 4 patients. And these patients, 4 patients, are patients that have been on gene therapy longer than any in the US. I'm just curious about how long you're going to be following them and how you're going to be measuring durability with these patients. Is it functional or measuring microdystrophin?
Yeah, great question. So the last data that we showed was at 2 years in the fall, and what we saw was durability in terms of functional improvement. So we saw continued functional gains from baseline, and that were consistent across time. So we're going to continue to follow them as part of the protocol for 3 years, and then an additional 2 years, so a total of 5 years. But as you know, we'll be continuing to follow these patients for quite some time and measuring their function over time.
Okay.
In the 102 study, we are measuring durability by having multiple biopsies, and so that study will be ending this year.
Okay, great, and great to see studies expanding into older. Okay, our next panelist is Beth Beluccio from Pfizer. Beth, I'm going to hand it over to you.
I'm Beth Beluccio. I'm the Global Clinical Lead for Rare Neurological Diseases at Pfizer, and hope everybody's ready for summer and enjoying this great conference. Really appreciate a chance to talk to you guys about our gene therapy program for Duchenne muscular dystrophy. Next slide. Right, so this also, we have a disclosure slide, but we can go on to the next one. So what I want to share with you today is an update on our set of studies to evaluate gene therapy for the treatment of Duchenne. Our therapy now has a new generic name. It's long, fortadistrogene movaparvovec, quite a mouthful. So from now on, I'll just call it by fortadistrogene. We do have 4 separate studies that will evaluate this therapy in Duchenne. We have our ongoing phase 1b study that now includes both ambulatory and non-ambulatory participants. We have an ongoing phase 3 CIFRIO study for the evaluation of ambulatory boys between the ages of 4 and 8. And then we have 2 planned studies, a phase 2 study for early symptomatic kids between the ages of 2 and 4, and a planned phase 3 study for non-ambulatory participants for which there's no age restriction. So during this presentation, I'm going to give you a brief update on each of these studies. Next slide. So starting with our ongoing phase 1b study, as you know, we started it in the ambulatory population, and we've now treated 19 participants. The age of the included participants so far has been between the ages of 6 and 12, with a median of 8 years old. We'll next update the community on data from this population once they've all completed 1 year since dosing. Now, we've also opened the study to non-ambulatory participants, and we took this step because we felt it was really important to begin evaluating safety and efficacy in older and bigger people with Duchenne. Ultimately, of course, our goal is to collect the information necessary to get this therapy approved and available for the entire population. So far we've treated 2 non-ambulatory participants in the cohort. One of them did experience a complement-mediated SAE that was very similar to what we've seen and reported in our earlier in our ambulatory population. So I want to emphasize about this that at Pfizer, we're really dedicated to the safety and well-being of these participants. And one thing that's clear about the studies is that we've already learned a great deal about what to look for these patients and how to manage the signs of complement-mediated immune response. So in this non-ambulatory participant, we were able to catch the signs early, and with treatment, he made a full recovery. So then I wanted to tell you also about a new cohort that we're about to open. This is because we're interested in evaluating sirolimus in conjunction with gene therapy. Sirolimus is a well-known medicine, and it may be an additional way to reduce the immune response to the AAV vector. You know, in Duchenne patients, we're all familiar with using steroids to inhibit inflammation. And what we want to do is understand if sirolimus could be an added tool to manage immune reactions. Sirolimus is also being evaluated by other companies in the setting of gene therapy for other disorders. So this is sort of an interest across the field. We will continue enrolling our non-ambulatory participants into this cohort as soon as our protocol amendment is approved, which we expect to be in the very near future. So what's really important about our experience so far in our phase 1 study is our data continue to support our chosen dose, and that also we have a really good process for monitoring and addressing any issues that may arise. So now I would like to turn to our phase 3 study in the next slide. So this is our CIFRIO study in the ambulatory Duchenne population, and you probably know that we had our first patient dosed last December. We continue to open sites around the globe. We now have 20 sites enrolling across 9 different countries, as shown in the map. Unfortunately, we don't have sites yet open in the US, and I know this is extremely frustrating to our families here. And so what I wanted to tell you is that we are working very hard at Pfizer to address the FDA requests, and we are poised to open our US sites as soon as we get the green light from the FDA. At this time, I can't tell you exactly when that will be, but I can promise you that we are doing absolutely everything we can to meet their requests. Now I want to tell you a little bit more about our 2 planned studies. So then in the next slide, describes our phase 3 global randomized double-blind placebo-controlled study in the non-ambulatory population. Similar to our CIFRIO study, we have a provision that all participants will be eligible for gene therapy, including those who are initially randomized to placebo. And also similar to all of our studies, we follow all participants for 5 years after they receive their gene therapy. Now, in this study, the inclusion-exclusion criteria will not have any age limits, but we will ask patients to meet some functional requirements, in part to protect patient safety while they're in the trial. Our primary endpoint will be the Performance of Upper Limb 2.0 total score, and we'll also very carefully assess both pulmonary and cardiac function. The full details of the study design will be available on ct.gov as soon as we get closer to starting the study. Next slide. And I also wanted to tell you about our phase 2 study in children between the ages of 2 and 4 with Duchenne. So this will also be a global study and include sites within the US. It will be a single arm study, meaning no placebo arm, and it will be primarily focused on safety. We will also evaluate, of course, efficacy in this age group, but endpoints for younger children are not established or validated, so we call them exploratory. We also will include muscle biopsies in this study to make sure that the mini-dystrophin expression is the same in this age group as we've seen in the older kids. And similar to our other studies, we'll follow all these kids for 5 years after they receive their gene therapy. Next slide. So just to conclude, I've told you that our phase 1 study is ongoing and currently focused on enrolling non-ambulatory participants with a new cohort soon to open for the assessment of sirolimus, and that our data continue to be very encouraging. We have good processes in place to maintain patient safety. Then our phase 3 CIFRIO study is enrolling with 9 countries open and more sites opening every week. And then we have 2 planned studies to evaluate our fortodistrogene in the non-ambulatory population and in children between the ages of 2 and 4. Next slide. So with that, I'll just end by thanking PPMD for this opportunity to talk about our program and also for their fantastic collaboration and support over the years. Also acknowledging the wonderful community of advocacy groups that we work closely with. And finally, I just want to emphasize that we also are very grateful to study participants and their families who are willing to take on the very hard work and stress of being involved in these clinical trials. We are really, truly appreciative to all of them. Thanks, and I'll take any questions.
Thank you so much, Beth. I'm going to just do a brief question and apologize to you and Carl for the quick switch and to the control room. So,
No problem.
So with the study that's enrolling now, both outside the U.S. and eventually in the U.S., could you explain competitive enrollment, and is there a possibility that the trial could fully recruit before this actually gets to the U.S. to recruit patients?
Right. So competitive enrollment really means that it's our intent to try and gather the data as quickly and as efficiently as we can, so that then we can be able to provide that to regulators. Because as everybody knows, ultimately the way you serve the entire community is by getting the drug approved. So we are trying to enroll as quickly as we can. And although we hope that we are going to be able to get U.S. participants in the study, there is a possibility, depending on the FDA response, that we will fully enroll before we get to that point. So that's why we're working so hard and diligently with the FDA to make sure that we can get the go-ahead as soon as possible.
Thank you. I appreciate that answer. Okay, we're going to change now to Carl this time. Carl Morris from Solid Biosciences. The floor is yours.
Great, thank you. Hey there, everyone. Yeah, so I'm Carl Morris, Chief Scientific Officer here at Solid Biosciences. Just want to thank PPMD really for the opportunity to present to the community about our AAV microdystrophin gene therapy program, which we call SGT-001, and our ongoing clinical trial, which we describe as Ignite DMD. Next slide, please. Not to make too much of a hullabaloo about everything, but as a public company, we have to share these slides, and please note our forward-looking statements here and on our website. Next slide. Yeah, so as many of you are aware, Solid was founded by parents directly impacted by Duchenne. So patients are really at the forefront of what we are trying to do. We really have engaged in the community, with the community, and truly value the input from everything from designing our clinical studies to providing meaningful insights into to further our understanding of the disease progression. Duchenne is a disease that really requires multiple perspectives to affect long-term changes and really to support development of potential treatments for this disease. So we work closely with people outside our walls to collaborate and identify ways to really drive our core mission in Duchenne. Internally, we've built a good team with vast experience in muscle biology, gene therapy, clinical and regulatory experience, as well as the patient advocacy that allows us to really sort of help develop those treatments for musculoskeletal diseases. And importantly, given the data that we've seen, the durable and so long-term data that we're seeing from SGT-001 really sort of supports the continued advancement of our clinical pipeline. Next slide. Over the past 6 months or so, we sort of announced the addition of two programs that provide sort of alternative approaches to deliver our microdystrophin gene therapy to patients with Duchenne. All three of these approaches use our differentiated nNOS binding domain containing microdystrophin, but each use capsids that may provide more opportunities for more patients to receive the payload effectively. For SGT-001, it's in our ongoing clinical trial, which I'll sort of talk to mostly in the next couple of slides. SGT-003 uses a next-gen, rationally designed capsid that enables us to express about 3 times more microdystrophin in the muscles of MDX mice at a similar dose to that of AAV9, which is our current capsid. So what we're hoping is that this will provide an opportunity and to have greater potency in delivery of our capsid transgene. And finally, the partnership with Ultragenics is sort of also enabling the use of an AAV8 variant and really a different manufacturing platform to help support getting the drug to as many patients as possible, as soon as possible. Both of these programs, the 003 and the Ultragenics programs, are quite early on in development, but we hope to, we at Solid will hopefully update on the 003 program when possible, and also Ultragenics will sort of begin communicating when appropriate. Next slide. So just to sort of set what we're doing in Ignite DMD. So currently, really our focus is on ambulant patients 4 to 11 years of age, although our first patient was a non-ambulatory 14-year-old, and we hope to get back there as soon as possible. When we originally initiated Ignite DMD, it was designed to be a randomized, controlled, ascending dose trial, and we subsequently modified it to an open-label, non-randomized design when we transitioned into the high-dose cohort. Today, 3 patients were enrolled at the low dose, 5 at the high dose, and a total of 3 patients were analyzed as part of the control cohort. The study population and entry criteria are listed here, and it's generally sort of a standard fare, antibody negative, and sort of a stable dose of corticosteroids is necessary. The primary endpoints for this study obviously is a phase 1/2, where with safety, as well as we looked at changes in microdystrophin expression in the muscle biopsies at 12 months, as assessed by Western blot, and I'll talk about that in a bit. As we reported in March, we resumed enrollment in Ignite, and we've now dosed 2 additional patients under the amended clinical protocol and with a new manufacturing process that enhanced our full empty ratio up to 90% full. We previously also reported, and so patient 7 was dosed uneventfully, while patient 8 was found to have a systemic inflammatory response and classified as an SAE by the investigator. Happily, and the patient has fully recovered, and all the lab values are back to normal. We shared the data with the DSMB and FDA, and we're working closely with both to sort of, move forward and get back into the clinic as soon as possible. Now to highlight some of our efficacy that we've sort of described. Next slide. Here, just showing one example of what we've observed. We were able to recently sort of take muscle biopsies in patients out to 24 months and then analyze them. And as you can see here in all 3 patients in the high dose cohort, that even out at the later time points, we're getting a good positive widespread distribution of microdystrophin positive fibers. And so out to 24 months in patient 4 and 12 months in patient 6. So we're looking at levels of 10 to 30%, up to 85% positive fibers in these patients. Also, we did look at Western blot to get a more quantitative assessment, and at the later time points, patient 4 remained sort of at below the level of quantitation, which is 5%. However, there seemed to be an apparent increase in expression from about 20% in patient 5 up to 70% of normal, while patient 6 I went from about 8% to 20% expression at the later time point. Next slide. So in March at the MDA clinic, we reported the long-term clinical biomarker and functional data is sort of highlighted here. As you can see on the heat map, there were positive trends in many of the endpoints in the dosed patients at that 1 year time point, with more green seen below, which is a positive response, seen at the bottom in the high dose cohort than that in the top in our control untreated group. The endpoints evaluated range from the creatine kinase, which is shown on the left, through several functional measures, North Star, 6-minute walk, and forced vital capacity, pulmonary respiratory measure, as well as looking at sort of an outcome measure, and all sort of trended in a really nice direction. For this 2E14, and at the time this was encouraging. Now with the promising long-term results that I just shared, it really sort of provides a mechanistic underpinning for what the apparent positive trends we're seeing here. Next slide. And so to summarize, just highlighting here some of the milestones and priorities for Solid at the beginning of the year. And really, we've accomplished 3 at present, as shown up top. So really sort of get back in and resume dosing of patients, identify some of the long-term safety and efficacy results, as well as sort of expansion of the pipeline. Importantly, and coming up soon will be, sort of evaluation of the 90-day biopsies from these additional patients, as well as sort of a discussion about how to advance towards commercial readiness and the preparation for our registration trial. And next slide. And finally, I'd just like to again thank the community, the patients, as others have said. It's amazing how the community sort of has been getting behind us, and as we sort of work to look for treatments to patients with Duchenne. Thank you.
Great, thank you so much, Carl. Just a very quick question. We all know this is a heterogeneous population where age might not be the best inclusion criteria. Will you continue to recruit based on weight and function rather than age going forward?
So right now we took a cautious approach and we were sort of reducing the weight for safety aspects and thinking about the total viral load. As we go forward, we will reconsider that and look about it, and maybe function, maybe age, as I agree with you, is not the best representation of how we should be looking at this, right?
Great, thanks. Now Eric's going to start the panel discussion.
Yeah, we've had a lot of questions come in. Many of them can go to the whole panel, so we're going to probably rapid fire around a little bit as we go through some of these. Obviously, one of the big topics and concerns is about neutralizing antibodies, whether that's pre-existing and preventing families from being able to access therapies, or for those who have been dosed and preventing the redosing. And so I think families would really appreciate hearing how all of the companies are thinking about this. What are you doing in terms of preclinical research or partnerships with other companies in terms of exploring ways that we can get around this challenge? And so Beth, I think I'm going to just throw it to you first, and then we'll kind of go around.
Yeah, sure. Of course, it's a very important topic, and it's going to be important for all the science to advance so that everyone can have ultimately access to the therapies. At Pfizer, we do have several sort of approaches that we're exploring in terms of how to reduce the antibodies, either to allow for redosing or to allow those who have existing antibodies to be treated. I think what our feeling is that at this point it's too early to tell exactly which approach will work. So we do think it's important to kind of advance on multiple avenues of trying to achieve that. Those studies are still very early. None of them are really ready for the clinic. But we are working to get ready so that we can advance them all to the clinic as soon as possible.
Thank you. Louise, could you share a little bit about how Sarepta is thinking about this?
Or this is a great question and something that we're very much dedicated to, both treating patients that have pre-existing antibodies and then for redosing. And so we have internal efforts that we're working on with ways to physically remove antibodies using things like apheresis that we've been working on for quite some time. We also have partnerships with Hansa that would potentially reduce antibodies in patients with pre-existing antibodies, and then select Biosciences at preventing antibodies and then potentially allow redosing. All of these are in preclinical studies in small and large animals, and we're moving them forward as fast as possible.
Great, thank you. And Carl?
Yeah, I mean, the same thing. We have a number of programs, and clearly this is a need, and we need to be able to dose as many patients as possible, regardless of seropositivity. And so, obviously, we're working hard, having an immunology team that's really focused and dedicated to identify either appropriate paths or partners to get this moving.
Great, thank you. And then another question. So you all have kind of different thresholds that you set for those antibody titers for when it's acceptable, in your eyes, to dose a patient. Can you maybe just talk a little bit about the evidence and the rationale used to come to what those thresholds are? Is this based on preclinical data? Is this learnings from other diseases? Carl, maybe I'll start with you and go from there.
Yeah, I mean, some of it's based on preclinical work and looking at what thresholds allow for transduction to continue. You don't want to lose your efficacy while you're dosing. There's both the safety side and the efficacy side that we have to consider in terms of the antibody levels. There's also some safety. We did some preclinical work and talked to other people in the preclinical space to really think about where a threshold may be in terms of safety. And so we selected that based on as much data as we could. But it's still a TBD in many ways.
Great, thank you. Beth, Louise, if either of you have any additional comments, you can throw them in. If not, I can get another question going. All right. So this one actually for both Sarepta and Pfizer. So you're working with and kind of preparing some placebo-controlled trials, and that does raise a challenge of there is going to be a period of time when patients are enrolled, not able to receive gene therapy. Have you had discussions with regulators about kind of the ethical aspect of that and potential ability for someone to develop antibodies while they're kind of in that waiting period? And maybe Louise, since Sarepta has done a crossover study, maybe start with you.
No, it's a great question, and it's not something we take lightly, but we have had those discussions with regulators, and the advantages after 1 year, the patients do cross over, even though we're mindful that is a year. We were grateful for the first 102 study where we did monitor patients and crossover. We did not see any patients seroconvert during that time that were unable to receive the therapy. So that was good to see, and I think it speaks to further work that needs to be done to make sure that we're being mindful about how seropositivity will, the chance of seropositivity or converting over a period of time. Sorry.
That's all right. Thanks.
Fumbling over my words.
Beth, can you maybe share as well any conversations you have all had?
Yeah, we've talked a lot with regulators about this because we really wanted to, as much as possible, limit or even avoid, if possible, the exposing kids to placebo and making them wait. But the regulators were really quite insistent on having fully placebo-controlled trials. And I think one reason is when they get the data, they want to be able to make a definitive decision and feel confident in the data so that they can hopefully approve these therapies. So we've talked to them a little bit about how to help protect patients who are then having to wait for the crossover, and we've also worked a lot with our experts and our investigators on trying to minimize the risk for the patients in the study. So it's great to have sort of that community of people really thinking hard about, if you do have a placebo-controlled study, how you try and make sure that patients will be able ultimately to receive that delayed treatment.
Great, thank you.
Great. Yeah, I have a question about, we're going to stick on this neutralizing antibodies question regarding screening. So collectively, we're getting close to, I think, around 100 patients that have been dosed, but even more have been screened for gene therapy. Are you able to comment on the percentage of screen failures based on neutralizing antibodies? I'm going to start with you, Louise.
Sure. Now, as we did a study looking at pre-existing antibodies for RH74 vector across DMD and limb-girdle, and in DMD, it was 15 to 70%, and the screen-out rate actually in our trials was less than that. So that was across a broad population that was not destined for enrollment. So it's actually been a little bit less than that for our trials.
Okay. Beth?
Yeah, so one of the things that we're sort of interested in gathering the data, although at the moment it's sort of premature, is how these rates, if the rates differ from across different regions, and if so, if there's any explanation we can get for that. In our global study, it's still early days, so I don't think we can comment yet on that. But it is something that we're interested in, sort of observing how consistent it is, or if there are regional differences in antibody levels.
Yeah, it's going to be important to publish on that. We're encouraging publication so we can learn from that. And Carl?
Yeah, I mean, again, similar to Beth, we use the same vector, and it depends on the stringency of the assays that you're using. But as reported preclinically, sort of the 30% level is sort of reasonable. 20 to 30% positive is sort of a reasonable idea of what's out there in the population.
Great.
And as you are all starting to think and plan for studies where you're looking at older patients, non-ambulatory patients, where we may have heavier patients involved, can you kind of talk to us about how you're thinking about weight-based dosing? Is there a threshold, either of weight or virus, where you kind of won't go over in terms of delivery, just from a safety perspective? And Beth, I think as you guys are kind of planning for this.
Yeah, I'd say that. One thing that we don't really understand yet is if there are going to be any effects that are related to weight and the total viral dose. It's a good hypothesis that there may be, but we don't really know that yet. So the approach we're taking at Pfizer is to sort of be cautious about starting with lower weight kids and building our experience that way. And then also, as we do that, trying to collect as much information as we can about other aspects of the patients, or their background or their disease state, that may also influence their reaction. So we are thinking about the weight, but trying to collect a lot of data to better understand, how the therapy may play out in the older population.
Great, thank you. Louise?
It's similar. I think, one of the things that was so important to us about expanding our 103 study for older and non-ambulatory patients was to gain that safety experience prior to starting the phase 3 study. And we're taking an approach where we're incrementally going to higher weights to gain that safety experience. So I agree, it's a hypothesis, but so far, collective data we have also from our other limb-girdle programs using the same platform, we haven't seen a weight-related adverse events.
Great, thank you. And these last couple minutes, we'll do some specific ones. So, Carl, can you give us a little bit of insight in terms of, as you're developing this SGT-003, but a question about maybe timing, and is this something that, if ready for clinic, would kind of phase out SGT-001?
So it's really early. It's sort of and we have to go through the necessary steps in drug development to get us where we need to get. The goal is to, as quickly as possible, sort of identify some clinical proof of concept. And sort of once we have those data and better understand what it gives us and what it doesn't, we'll be able to make sort of really those types of decisions as to whether we're going to be, is it taking the place of SGT-001? Is it additional? Does it provide some other differentiated function that we're just not aware of yet?
Okay, great, thank you. Louise, could you possibly shed any light on the difference between that study 301 and 303 that you showed kind of at the end of your slides?
Sure. Yeah, 301 will be more similar. We're waiting for feedback from FDA on a START 1 or 2 study in the 4 to 7 year old age range, and then the 303 will be older, non-ambulatory.
Okay, great, thank you.
Mhm.
And then the last question, Beth, can you give us any update on your progress with that potency assay with the FDA? And maybe just also a brief explanation of what that is for families who may not be aware of potency assays.
Right. So, so the FDA, of course, looks at lots of aspects of clinical development, and they look at our studies and how they're designed, and they also look at how we make the gene therapy compound, so the manufacturing process. And they have every company, Pfizer, and I'm sure the other companies have a long list of tests that we put the compounds through before we give them to patients. Every single time it's manufactured, you run the compound through these tests. The potency assay matrix is just one of those tests. And the idea there is to make sure that the compound as it's manufactured will have approximately the same effect as another, that every vial will have approximately the same effect. So the FDA is working with the companies to make sure that the tests that we're using match what their specifications, and so what we're doing is just working to try and meet the requests that the FDA has put into place in terms of what they want those tests to be. And it's an ongoing conversation, and we're very engaged with them. We have confidence that we'll be able to meet their requests. So we're just working on that as quickly as we can.
Great, thank you. And thank you all for joining us today. We really appreciate you taking the time and discussing some of these questions. There's a lot more for us, obviously, to dig into, and I think this will create some good education opportunities in the future. So thank you all for joining us tonight.
Yeah, that's great. Thank you, everybody. And the
Thank
good
you.
news is that many of these companies are going to be involved in our update to the Duchenne guidance. We're sort of going to force them to work together and talk through issues that could help as we develop gene therapies. So we're excited for that. So now we're going to hand it over to Pat Furlong. We are so lucky to be here today with Dr. Celia Witten, who's joining us. She's the Deputy Director at the FDA Center for Biologics Evaluation and Research. It's much easier to say CBER. This is the center at FDA that oversees gene therapies. We've known Dr. Witten for many years. We've had the pleasure of working with her, and I'm so grateful that she's here to talk with Pat. Pat, I'm going to hand it over to you now.
Thank you, Ryan, and welcome, Dr. Witten. Thank you so much for joining us tonight. We so appreciate your time.
Thank you.
Sure. I have loads of questions for you, Dr. Witten, in our short period of time. The first one is about CBER and CDER, the Center for Drugs and Center for Biologics. Our community is very familiar with CDER, the Center for Drugs, and now very hopeful for gene therapy, which sort of lands in your lap. Is there a crosstalk? How do you communicate and get all of the learnings that have occurred in the antisense oligonucleotides and the steroid approvals and take that into account as you look at gene therapies?
Thanks for that question. We're very excited about gene therapy in this area. I'll just say that. And I think our communication with CDER, who has more, more experience with approvals, is important. But we've had regular communication for years. So I'll describe some of the forms of communication. They are both programmatic and also in the setting of specific applications. So we have a monthly meeting that actually includes all three product centers, CDER, CDRH, and CBER, to discuss specific neurology applications and topics of shared interest. So those could include things like clinical issues, endpoints, trial designs, and also the rationale between specific regulatory decisions. So that is an important forum for communication, so we have regularly know what is up in each other's area and can have some discussion about it. And also consultation is obviously important. So when we review specific applications. We frequently consult other FDA centers. In this case, it would be the neurology group at Cedar, and their perspective, expertise, and experience are often useful in our consideration of clinical issues like the interpretability and meaningfulness of proposed endpoints. And then there's some other efforts in terms of, development of tools for product use that I think are still in development. We don't really know, how they'll be used, but we certainly are interested in the, the CPATH efforts and the area of drug development qualification program. So we do learn about those and follow along and hope to benefit as those, tools evolve.
Yes, thank you. We had a call with Krista Vanderborn from Imaging DMD, and she's talking about the qualification process for fat fraction. So we're very interested in those tools. So Duchenne is a slowly progressing disease, as you know, and sometimes it's really hard, especially in short periods of time, to evaluate safety and efficacy. So I would love for you to comment on this accelerated approval pathway that we experienced in the Center for Drugs, and how you're thinking about that in gene therapies.
Yeah, so just, to take a step back and talk about just the overall context of accelerated approval. And I agree it's an important topic. Home runs are easy to assess. Of course, that's what we always want, would want, hope for. And if you have one in Duchenne's, for example, maybe you'd see the disease stop, the strength return to normal, a normal or a near normal biopsy. And that would take only maybe a clinical trial of a year or two to be confident. But home runs are easy to assess. But the challenge is that most products are not home runs, and the benefits may be more modest. The interpretability of the study then becomes more subjective and variable. So in these situations, just as a general matter, it becomes critical to have concurrent, controlled, appropriately blinded clinical trials, and longer duration studies become even more important to be confident that the effect is real. And I just caught the tail end of the previous discussion, so I know there was pretty active, comments on some of these points. But we support CBER as a center, certainly supports accelerated approval, and the office that regulates gene therapy for Duchenne's in particular used it recently in the approval of a CAR T-cell product in oncology. So it is part of what we do. Accelerated approval relies on a surrogate endpoint that's reasonably likely to predict a clinical benefit. And our ability to rely on such a surrogate depends on having a good understanding of the pathophysiology and natural history of the disease and the role of the proposed surrogate in that pathophysiology. So for many rare diseases, those may not be well understood. Fortunately, in Duchenne's, the pathophysiology is generally well understood. The role of dystrophin in that pathophysiology is well understood. In gene therapy applications, it should be noted that the sponsors have proposed to use mini versions of dystrophin as a surrogate, and the role of mini dystrophin in the pathophysiology of DMD is less clear than the role of dystrophin. I'm just mentioning that is one of the things that we would keep in mind. So in general, though, just getting back to the general topic, if a drug company proposes to use a specific endpoint as a surrogate for clinical benefit, we would ask them to submit data to support that the proposed surrogate is reasonably likely to predict a clinical benefit. So we're always open to this option, but we need to see data to support the proposal. So I guess
Sure.
that's right.
It's hard, right, because of the slow progression, and I do understand. And so as you look at, you're doing many, or you're overseeing many gene therapy, pediatric gene therapy studies. What are the ways you're thinking about to accelerate approvals or to accelerate this process? As you know, so many people are out there waiting and hoping for certainly a gene therapy that is both safe and effective. So how are you thinking about that?
Well, that is a really good question. And let me say that one of the things that we care about and always are thinking about are any efforts to develop cross-cutting tools. and something that might be useful in more than one setting, that is not in a specific sponsor's own development program, but across the board. And so one area, as you know, that is definitely of concern to FDA and the community and the patient community also. And again, I heard a couple of questions about this in the tail end of the last discussion, is the question about neutralizing antibodies and neutralizing antibody testing. And so that is, it's important. And it's important to understand what the results mean. And it's important both for the individual product, but it's important across the field to really, understand it so you can interpret things from one setting to another and really understand, the data more generally than just what the results meant for that setting. So we have, for that kind of thing, and for a number of other topics like that, a couple of years ago, FDA stood up a group, an organization. It's a public-private partnership called the Standards Coordinating Body. And that organization looks at opportunities for standards development in the field. This is one of the topics that they looked at. That group doesn't develop standards, but they support standards development by facilitating participation of U.S. experts in standards development efforts. So I'm hoping that this topic of neutralizing antibody it is proposed as one of the topics at an international standards organization to take up. And what that means is they would develop test methods that people could more uniformly apply for that kind of testing, so you could interpret it better across the field. And there's a lot of examples like that. So the standards world is one area where I think further work in standards can enhance the field. Then there's other questions like, how do you leverage information from one development program to another? And that could be the data about the vectors. It could be information about preclinical testing. I mean, it's a general question. So one of the things that CBER is doing is partnering with the Foundation for NIH, NCAT, and NCATS on the so-called Bespoke Gene Therapy Consortium, BGTC. And the objective of that group is to look in a consortium framework at a common set of procedures in a pre-competitive environment to see whether that can drive innovation and promote access to individualized gene therapies. And it's hoped that we will learn something from that effort that will, the phrase "a rising tide lifts all boats," that in that effort to figure out how to develop, test, and deliver therapies to very small diseases, we'll learn things that'll help with larger development programs in the community as a whole. So it can be questions like, how do you leverage preclinical and CMC data, or learn about innovative trial designs that might facilitate development, and even simple things like the use of master files, procedurally, like what's the most efficient way to help manufacturers submit, those who have a platform technology. So those would be some examples of what we're doing. And maybe that was too long-winded, maybe.
No, it was wonderful, and we can't thank you enough. I think that neutralizing antibodies, for patients and families to understand what the testing means and whether it applies to all serotypes would be impressive. I think how to handle the complement response when these children get a virus and have a response to that, and if that could be generalized, and certainly data and looking across. I think all of these things are so important for our families and for the world of rare diseases. I can't thank you enough, Dr. Whitten, for what you do, for how you communicate with all of us and how you're working so hard. If there's anything we can do to help you accelerate, our fingers are crossed and we're hopeful to get a home run or even a near home run in gene therapy. So thank you for joining us tonight, and we hope to see you very soon.
I appreciate that. And I just want to add one last word, which is we appreciate the efforts of the patient communities, and we're looking forward to seeing the guidance document as you develop it.
Thank you. Thank you. We're thrilled to be developing that guidance. Thank you again. We'll see you soon.